Hi, my name is Dina Finan, and I'm a product manager at 10x Genomics. Today, I have the privilege of giving a short introduction before sort of an overview of our technology before my colleague, Stephen Williams, takes it away to tell a story with some interesting data. So I'm sure you're all no doubt well aware that human biology is immensely complex. A significant amount of research has obviously been done already to uncover all of these complexities, but there's still just so much left to discover. So from the trillions of cells that make up our bodies, encompassing many different cell types, the genes encoded in our DNA, the structure of the chromatin, and the transcription and translation of all the genes, depending on the cell types, there's just so many different layers of complexity there. So at 10x, we developed tools to try to resolve this complexity, and it's our mission to continue to push the technology boundaries to offer solutions to accelerate your research around this. Our solutions really span the whole process, as depicted in the slide, from the assays and the biochemistry behind them, to the instruments, down to the powerful analysis and visualization tools in terms of software. So the core of the way we address this complexity is to use barcoding, and that allows you to maintain the identity of single cells, single regions of interest on a tissue, or single features, while still allowing you to process a lot of samples in bulk. So even before the last couple of months, where it's become painfully clear to the world how important it is to gain deeper understanding of biology, the last few years have really just been a golden era in terms of speeding up discovery across life science. So single cell sequencing has actually been an engine that's driven a lot of this research that's been published in a lot of top-notch journals over the last few years. In 2019, we actually saw more than one publication per day using our solutions at 10x for a total of over 700 papers. And these publications really cover an incredible range of research areas, from oncology to immunology, developmental biology, and neuroscience, among others. So to give you an introduction or a recap, depending on your familiarity with our workflows, for single cell work, the carefully integrated instrument, the consumables, and the reagents, they take you all the way from a single cell suspension, your sample, to sequencing ready libraries. And on the other end of the sequencing, the analysis and visualization pipelines take you from FASTQ files to reduced data and give you some biological insight into those samples. So as I mentioned earlier, with the number of publications and just all of the good research to highlight, it's very difficult to choose just a couple to focus on. So these two papers are just among my current favorites. And so I just wanted to kind of bring them up because they both tackle really complex diseases. So the one on the left is out of UCSF, and the one on the right is out of the Broad Institute. And both of these are, like I said, tackling extremely complex diseases, autism and Alzheimer's disease. And they're both case-controlled studies that look at cell type specific changes in gene expression to get a much more detailed picture of the changes that occur in disease-affected tissue. So the types of studies that are done here, I think, really highlight, if you aren't sure the why to do single cell research, this is really the three reasons I think that are highlighted here. So number one, you actually learn the cell type that you're looking at where the change in gene expression occurred. So that's really critical information. Number two, you increase your statistical power because you're comparing expression levels of cells of the same type, which makes a lot of sense, right, when you're looking at gene expression in a complex tissue like the brain, for example. And then the third thing is you have the power to distinguish between changes in expression levels versus changes in the abundance of cell types. So these are a couple of reasons that I feel like if you haven't considered some of the reasons to do single cell gene expression studies, these papers might change your mind. So our newer Visium platform follows a similar template as our single cell. And so we leveraged our experience with the single cell work to dramatically improve the sensitivity and the ease of use of the spatial transcriptomics platform. So here we adapted our pipeline and our visualization software as well to be able to integrate imaging and molecular data on top of the sequencing data. And Stephen's talk, I think, will really highlight some of the advantages of being able to do this. Oops. Okay, so over the last 5 years at 10x, we've introduced a whole suite of different applications in different areas. So I've talked a little bit about the single cell gene expression, and I wanted to highlight just a couple of others to call attention to them. So our immune profiling solution allows you to highlight, to simultaneously examine the cellular context of the adaptive immune response and the immune repertoires of anywhere from hundreds to tens of thousands of cells at the same time. So you can look at T cells and B cells, cell by cell. And with the feature barcoding technology added on top, you can also look simultaneously at other kinds of cellular readouts, like cell surface proteins and antigen specificity, which is very powerful. And then our ATAC-seq in green over here in the middle of the 2018 group, the ATAC-seq solution enables you to look at chromatin profiling of thousands of single cells in parallel, so that to kind of advance the understanding of how chromatin compaction and DNA binding proteins regulate gene expression at very high resolution. So these are two really interesting things that have come about recently at 10x, and you can kind of see how rapidly new technology is being introduced. And so with that, I also wanted to say that this year there's a lot more to come, and I think Stephen's giving a little teaser on something in the Visium platform. But I really encourage you to go to our website. There's this page/future, and you can sign up for updates to stay connected and find out what exciting things we're doing next. So please stay in touch.
Hello, my name is Stephen Williams. I am a senior scientist at 10x Genomics in the Computational Biology department. I want to thank Dina for the nice overview of the solutions that 10x Genomics has to offer and some of the things that we're working on down the road. Today, I'm going to give an overview of using Visium to spatially resolve triple-negative breast cancer. So first, I'd just like to give a small overview of the Visium technology. So the aim of Visium is to have unprecedented resolution, being able to capture simultaneous analytes and put them all in complete context, while at the same time doing this at scale and making it accessible and easy for every lab to run any 10x Genomics application. Fundamentally, the Visium spatial application is based on a slide that contains 4 capture areas. Each one of these capture areas contains 5,000 barcoded spots that have a partial read 1, a spatial barcode, a UMI, and a poly-DT to capture polyadenylated mRNAs. Each spot is printed in the exact same place every single time, so we know which barcodes go where, and this is what really enables being able to map RNA expression back to image space. So the spatial gene expression workflow goes as follows. First, you section your tissue on your normal cryostat that many labs and hospitals have. You do your Visium prep, which first you take a H&E image of your tissue, place it on your capture area, do permeabilization, and then all the mRNAs are then captured by the oligos that I showed in the slide previously. Then you do a standard library prep sequence on your favorite sequencer, probably Illumina, and then everything is kind of done for you from that point on. The Space Ranger pipeline takes care of all the tissue segmentation, tissue detection, and representation and visualization of all of your mRNAs in tissue space. And this is really enhanced by the Loupe browser that allows you to zoom and do differential expression and create custom clusters. But these outputs from both Space Ranger and from Loupe both can be imported into community analysis tools such as Surat or Monocle or ScanPy, some of the things that I'll actually show today. That's with the exception of the Loupe file. So Space Ranger is a Linux command line software that is used to process the sequencing data for downstream analysis. Automatically aligns tissue and gene expression data and detects spots under tissue and identifies spatial gene expression patterns, cluster spots to facilitate cell type annotation with downstream tools, and generates actionable QC metrics on these very nice HTML pages that we produce with every Space Ranger run. The Loupe browser is amazing, and it's got a real upgrade with the addition of spatial data. So Loupe is an intuitive desktop visualization software that allows you to interactively explore your data. And when I say interactively, I mean really interactively. You can zoom, you can pan, you can change colors, you can export your data, you can import data. It is really flexible, and it's become a very powerful tool to use in discovering the underlying biology of the experiments that you might run on Visium. But that's not it. So coming soon to the Visium platform, we'll be enabling targeted gene expression, immunofluorescence integration, and I'll show some of that today, feature barcoding technology for things like feature barcoded antibodies, and enabling solutions for FFPE. So our assay development team has done a lot of work on breaking down the tools and the processes that are needed to properly prep a variety of different tissues from human, mouse, and rat, actually. And you can see there are a wide variety of human tissues as well as mouse tissues. But the one that I'll be speaking about today is breast tissue in the context of triple-negative breast cancer. So now I'm going to just give a brief overview of triple-negative breast cancer. So triple-negative breast cancer is a devastating disease, as many of you know. But the specific phenotype for triple-negative breast cancer is that these individual tumors are negative for estrogen receptors, progesterone receptors, and excess HER2 protein expression. The burden of triple-negative breast cancer on our society as a whole is tremendous. This is a very aggressive disease that accounts for 10 to 20% of all breast cancer cases that are diagnosed in the United States annually. And what makes this disease very tough is that it's resistant to conventional therapies, which leads typically to a poor prognosis. So understanding the underlying biology and the microenvironment of each individual person's triple-negative breast cancer is essential to improve diagnosis and therapeutic outcome. So first, I'd like to give an overview of the molecular design of our study. We obtained two tumors from middle-aged female cases, and we purchased them from BioIVT. And these individuals are from diverse ancestry. They have high-grade tumors, so grade 3. And we did a lot of work on these tumors. We took serial sections and ran them on Visium, at least 3 sections for each version of the workflow that we did. First, we did the standard Visium workflow, H&E staining with follow-up with capture of mRNAs. Then we did each of these H&E stained images was sent to a pathologist who annotated them as well. Then we did integration with immunofluorescence using CD3 and CD8 protein antibodies. And I'll be showing some of the CD3 data today. And then we also ran these Visium samples on our targeted pan-cancer panel, which will be coming out later this year. I'm not going to show any of that information, but I can say that it worked great, and it's a fantastic way to lower your sequencing costs if you know what you're looking for. We also paired both of these tumors with single nuclei RNA sequencing data. So from each individual tumor, we obtained approximately 40,000 single nuclei, making a metadata set of approximately 80,000 single nuclei. And these were annotated by one of our expert scientists at 10x Genomics. So now I'd like to talk about our study overview from the computational perspective. So we did a variety of different things, a lot of which was custom, but all done with the basis being the standard Visium workflow. So we did differential gene expression in space. This was done with Space Ranger, not Cell Ranger. We did integration and correlation of clustering with Pathologist Annotations. These were imported into R and plotted on top of our Visium images. We used these to do custom clustering and differential gene expression. We did quantification of fluorescence intensities, and this was performed with R as well. We used these intensities to do custom clustering and differential expression in Loupe again. And this gives us insights into cell type specific behavior. We also used the SURAT package to identify spatially variable features or genes. And we integrated single nuclei RNA sequencing data with SURAT to identify cell type specific expression patterns in Visium. So now I'll give a intro to the Visium technology and our basic analysis using Space Ranger. So fundamentally, the Visium assay is powered by an H&E tissue section, capturing that mRNA, putting it through the Space Ranger pipeline, and outputting really nice information that you can explore in Loupe. And all of this is prepackaged in a very easy to use workflow. The typical output of a Space Ranger run will be you'll be able to visualize your H&E sample in space. You will then be able to visualize mRNA expression on top of your H&E image. And each one of these spots represents one of the printed spots on the capture area. And in the colors represents the total UMIs that are identified in that capture area. But then you can actually run clustering algorithms. We provide 11 of them, a graph-based clustering plus k-means 1 through 10. And this gives you information about what types of, what spots will cluster together by gene expression profile. So there are some very standard techniques in the field of pathology. And the very basic level of it is annotation by a pathologist, which is not simple. People get doctorates in this, but it's information that can be layered on top of your Visium data. So here are 3 sections from one of the cases of triple-negative breast cancer. This has been annotated by a pathologist. In the green area, kind of in the middle, you can see that they're marked with invasive carcinoma. In the lighter green area, which is a little bit covered up by the blue, you can see that they have annotated those regions as ductal carcinoma in situ. They've also identified regions of fibrous tissue, fat, immune cells, which are all extremely interesting. And to be able to layer this information on top of what you'll see in expression space, I think is really cool. So here's just the first section from this case. And I'm showing you how you can see the H&E image. You can then see the annotated image, and then you can see the clustering laid on, paired with that annotation. And what's really interesting, I think, in this visualization is that the clustering for this light green, red, and somewhat blue tissue or spot actually pairs up extremely nicely with what the pathologist annotated as invasive carcinoma. And these are the kind of images that I'll be showing from here on out. So please have a look at this and familiarize yourself. So next, I'm going to show how you can convert these pathologist annotations into something that can be ingested by Loupe. Because right now, you are not able to import the annotations themselves into Loupe. Rather, those were something that I did in R, but it's very easy to do. So you have your H&E image. You then have your custom annotations from the pathologist. But then you can associate each spot with one of those annotations. And you can see that I've done that in the right-hand side. Now, once you assign a spot, which has a barcode, to an annotation, that's just considered a custom cluster. So that is something that Loupe can ingest. And so when you put that into Loupe and load it up, you're then able to use Loupe to do differential expressions between your new clusters defined by fibrous tissue, immune tissue, ductal carcinoma in situ, and invasive carcinoma. And when we did that, we were able to see that we pulled out some really interesting genes, many of which are pretty heavy hitters in the field of breast cancer study. And it was really gave us some confidence in what we were seeing in the clustering versus what the pathologist annotated. And then you're laying on this whole nother information about what exactly is happening inside your tumor at the microenvironment level. So next, I'm going to talk about integration with immunofluorescence. So immunofluorescence has been a long established technique of being able to visualize protein expression on top of a tissue. So first, we took one of our breast cancer samples. We stained it with a CD3 antibody. You can see in red, that's the CD3 staining. And then in blue, you can see DAPI staining for nuclei. You can then take this image, and you'll actually take this sample and run it through the Space Ranger pipeline. And what you get out is spot information about your CD3 mRNA expression. So in this, we've been capturing, we just made a global signal for T cells because CD3 is very lowly expressed for mRNA. And this is actually where the power of using immunofluorescence and pairing it with Visium comes into play. So you're able to detect both things and layer them on at once. So you're able to also merge your protein data into spot space. So if you have your CD3 channel on your immunofluorescent image, you're then able to identify the protein intensity of each spot. Now, this isn't done in Loupe. This was done in R, but it's actually very easy to do. So you create a mask and essentially just detect what the light intensity is at each spot. We didn't do any fancy normalization or anything like that. But it is really powerful because then you can pair that and correlate it with your mRNA expression, while the CD3 mRNA expression for T cells is much lower than the protein. You can see that the patterns are very similar. So next, you're able to, just like I did before with the CD3 mRNA and classifying things into a high versus low CD3 expressing spot space, you're able to take your quantification of protein levels by calculating your intensity of light in spot space. You're then able to make custom clusters out of those. So here I'm just naming my T cell markers as, does this spot express a T cell marker in protein space or does it not? You're able to then import those into Loupe, just like I did with the custom Pathologist Annotations. And then you're able to do differential expression. And what you can see is that for spots that have high CD3 immunofluorescence versus low immunofluorescence, you can see that it is just chock full of immune representing gene expression profiles. And so that's completely expected, probably not something that's shocking, but it's a great validation of the things that we're doing down the road. So now I'd like to talk about identification of spatially variable features. And we'll get into kind of what that means in just a second. So typically you have your gene expression profile, and you can have your clustering represented in UMAP space, like I do have on the left-hand side here. And you can see that this is for the first section of case number 1. And you can see that you have nice clustering. You have spots that are tightly packed in TC or in UMAP space, and that indicates that they have similar gene expression profiles. You then can map that expression and clustering profiling back onto your image that went into Space Ranger, and you can see that you have some nice distinction of clusters within this space. The things that I'll probably pay most attention to here is you can kind of see the outline of a boot with the tip facing left. And that is where the pathologist has annotated this sample as ductal carcinoma. But what's really interesting is then if you say, okay, well, I identified this gene IGLC2, which is the constant region of immunoglobulin light chains. And in the recognition phase of hormonal immunity, this membrane-bound immunoglobulin serves as a receptor upon which antigens can bind and can trigger the clonal expansion and differentiation of B lymphocytes. So this would be a really nice marker of B lymphocytes. And it looks very interesting in UMAP space. You can see that it's expressed in these few clusters in red on the kind of lower left-hand side of your UMAP. But then when you plot this information back onto your tissue, you're actually able to visualize the expression in tissue space. And you can see this is probably where the majority of my B cells are residing. And so I think that's pretty cool. And I'm going to get into some even further expansions on this technique. So in addition to your typical differential expression, you might want to see what genes are actually spatially enriched. So we use the SURAT package and their function called Find Spatially Variable Features to search for features that exhibit spatial patterning completely in absence of any sort of pre-annotation, whether that be clustering, cell type, or anything like that. So this method is based on the TrendSeq paper, which was published in Nature Methods in 2018. And it models spatial expression as a mark point process and computes a variogram. And so this expression level is dependent on their spatial location. So it takes into account things, genes that are expressed in similar space. So when we ran this, so just to note, the input to this function is actually the top 1,000 differentially expressed genes. So you're kind of paring your gene list down a little bit. And that's really just to speed the process up a little bit. So here you can see that on the top row, we have 2 genes that are associated with immune response in cancer, CCL19, a cytokine, and IGHA2, which is another immunoglobulin. On the bottom row, we see two genes that are enriched in the region of invasive carcinoma. This migration and invasion enhancer 1, which is a known gene associated with cancer metastasis. And then this SLPI gene, secretory leukocyte protease inhibitor, which is upregulated in highly metastatic triple-negative breast cancer. And these cells typically secrete more SLPI. And so I'd like to dig into SLPI a little bit more. So SLPI was kind of deep in the weeds. So if you look at, it was not an obvious candidate when we ran just pure differential expression between clusters. It was actually ranked number 200 in our cluster-based differential expression experiments. So I don't know about you, but I probably would have missed this gene back in the days where I was doing a lot of this sort of research. But when we ran our spatially enriched gene experiment, SLPI was ranked number 15. And I would guarantee that this gene will be captured after this experiment. And it seems like this gene is an essential part of defining the microenvironment of this particular cancer. So on the right here, you can see I'm highlighting where the SLPI gene is expressed. In the upper left-hand corner, you can see these two circles where it's most highly expressed. Those correlate with this darker green clustering. You can see in UMAP space, that is correlated with cluster number 3 up in the upper right-hand corner of the UMAP. And then what I'll be getting into a little bit more down the road is that this actually correlates extremely well with what we define as ductal cells when we integrate the single nucleus RNA sequencing data. And this is where we're really taking things to the next level by not only seeing your gene expression information in space, but also being able to associate that space with a particular cell type. And that's what I'd like to get into next. So single nucleus plus Visium integration, I think, is something that everyone wants to do. And there are a bunch of groups working on it, and something that we want to enable. So why is single nucleus integration powerful? So there is just a massive wealth of single cell and single nucleus data that is being generated. Resources like the Human Cell Atlas, I think, will be invaluable in determining what makes a cell, just as the Human Reference Consortium was invaluable in mapping and defining the standard for a human reference genome. So Visium captures multiple cells per spot. It is not a purely single cell technology. And untangling these mixtures improves our ability to understand the tumor microenvironment, I think, is really the end game of what most researchers are interested in, because it's very all well and good to understand where and at what levels your genes are being expressed. But if you don't have that cell type information layered on top of it, you're missing a key piece. So the study design of this single nucleus RNA integration, and I'll briefly just state the reason that we did single nuclei is that we're using fresh frozen blocks. It's very hard to get single cells intact out of a frozen block. That is something we're working on, though. So these single nuclei came from the same tumor as the Visium samples. They're from the exact same block. We isolated these single nuclei. Like I said, we captured 40,000 nuclei per tumor. We then aggregated these samples together and using Cell Ranger Agar, and then we normalized them via Harmony. Clusters that were then annotated by our resident 10 expert scientist, Dr. Wyatt McDonald. And then we did the actual integration with SURAT. So SURAT uses an anchor-based workflow, which was introduced in SURAT v3, that enables the probabilistic transfer of annotations from a reference, which is your single nuclei, to a query set, which would be your Visium data. And you can read more about that on their spatial vignette that they put out earlier this year. So here is a UMAP representation of our clustering of our single nuclei RNA sequencing. So you can see we've got a large cluster of T cells, we've got B cells, we've got ductal cells, stromal cells. We also have tumor cancer cells, which are defined by this CD163 positive MPH. But then in the center, I'd like to draw your attention to our tumor cells, but also that they are very close in UMAP space to luminal progenitor cells. And I'll be talking about that phenomenon just a little bit later. So here you can see the integration of the single nuclei RNA sequencing data and identification of spots that are most likely T cells. And that's represented by this color scale, blue being less likely to be a T cell, red being more likely to be a T cell. On the top row, you can see that outside of what was normally annotated by the pathologist as ductal invasive carcinoma, invasive carcinoma, you can see that T cells reside mostly on the outside, which isn't surprising. But you can see you do have some invasion of most likely of tumor T cell infiltrating lymphocytes or tumor infiltrating lymphocytes. Now, you can also do the same with B cells. So B cells reside almost exclusively outside of the tumor microenvironment, or what's defined as purely invasive carcinoma. And that makes sense too by what we know in tumor biology. Something that's very interesting though is that we identified 4 different tumor subtypes, and those subtypes are regionally defined. So you can see that subtype number, what we're calling 3, that was the cluster that it was associated with, is kind of in the middle of the invasive carcinoma. So this is just one slice that I'm showing here. On the upper right-hand corner, you can see another tumor subtype, which is very nicely defined on the right-hand side of the invasive carcinoma. Then you've got these other tumor cells that seem to be spread throughout the tumor on the bottom left-hand side. And then you have another tumor subtype, which seems to be just kind of sparsely expressed, but regionally well-defined. So we've got these tumor subtypes, but what about how this tumor may have evolved? And so one of the methodologies to be able to identify how any cell type may have originated is to do a trajectory analysis. So we performed a simple trajectory analysis using Monocle and discovered some very interesting aspects of the potential development for this subject's tumor. So trajectory inference approaches analyze genome-wide omics data from many single cells and computationally infer the order of these cells along some sort of developmental trajectory. So I want to draw your attention to this middle group right here. So here we go. So in the black line, you can see this is actually a graph that is inferring what the trajectory may have been for this group of cells that are mapped in UMAP space. So at the node of this graph at the bottom, you can see, or one of the nodes, excuse me, you can see that this is luminal progenitor cells. And so they seem to be developing into these tumor cells at the end. And so deregulation of luminal progenitor cells are known to be the origin of basal type triple negative breast cancer. So I think that we've got a really good snapshot of how this tumor evolved from the very beginning. And subtype frequency associated with is associated with P53 function and MET signaling and hyperactivity. So this is a really interesting take and discovery for this single individual's tumor evolution. So these liminal progenitor cells, it's great to identify them, but where are they residing? So you can see on the top row of this Visium plot, you can see that the liminal progenitor cells really are residing somewhat on the outside of what the pathologist has annotated as invasive carcinoma. But I kind of want to draw your attention also to these kind of two circle bare spots in the middle where SLPI was expressed, and you don't see them there. So this is indicative that those regions probably have more mature tumor cells, and that makes sense. So I mentioned before also that we are super interested in the phenotype of tumor stem cells. And so it seems like these tumor stem cells, while somewhat rare, seem to be at the tip of the boot of the invasive carcinoma. And I have always wondered, since I've seen this data, could these be the key for targeting and therapy? And because, and the thing that I found really interesting was this entire region, this kind of tip on this boot, as you will, was defined as invasive carcinoma by the pathologist, but there seems to be a real mixture of cells going on down there. And could this be some sort of transition space or something that might be a gateway to therapy for this individual tumor? And that's something that we're really interested in digging into. So that concludes the section for integration with single nuclei RNA sequencing data. Next, I'd just like to kind of give a shout out to Rahul Satij's lab at New York Genome Center. They put together an amazing vignette on processing Visium spatial data. Myself and Nigel Delaney helped get this kind of off the ground from the beginning, but they really ran with it and have put out something that is typical of Rahul's lab in being an excellent tutorial. This tutorial can be found at this URL. It's their vignettes/spatial vignette. So in summary, we have shown today that the future of Visium brings a wealth of new applications, much more than what I showed today, as we're integrating a whole bunch of other applications. But we would really like to know what you need as researchers to achieve your goals in science. So feedback would be excellent, and I'd really encourage you to ask questions after my talk, which I'll field via email. And Visium enables the understanding of the tumor microenvironment at a new level. You can integrate protein-based assays, you can integrate fluorescence-based assays at the same time while capturing RNA-seq data in the same spot space on the same tissue. You can pair these annotations with Loupe for processing, so you don't have to do any heavy computational lifting. And we've designed these data types to be paired with real world-class third-party tools. Detection of spatially enriched expression patterns is one of the things that I'm super excited about, and I really believe that this can lead to new discoveries. And integration with paired or reference single cell or single nuclei RNA-seq data is essential for characterizing a variety of different organisms and disorders. So I'd like to thank our 10X team. It is a fantastic group to work with, and also all our outside collaborators, both in pharma and in academia. And at the very end, I'd like to just pitch a webinar that we're having on May 6th that's being held by Wyatt McDonald, who did the amazing cell type annotation for us. and he'll be presenting a talk called Revolutionizing Antibody Discovery for Pharma with Single-Cell Approaches and LibreSeq. And that could be, you can register for this webinar on our website at this URL found below. And thank you. I'd like to thank the organizers for inviting us. And I hope that this was an interesting insight into things that we're doing on the Visium team. And I will field any questions via email later this week.